How can bacteria make a human population evolve? What does cholera have to do with the blood types in the Ganges Delta? The answers to these interesting questions can be found in a paper published in Nature Genetics titled Natural Selection and the Infectious Disease in Human Populations. Let's start with the basics. Natural selection is a process in which the fittest individuals survive. Fittest is classified through certain genotypic characteristics that make them better adjusted to an environment. Being better adjusted leads to increased survival, reproduction, and ability to pass their genes on to the next generation.
The most recent studies on a large population scale use GWAS, or Genome-Wide Association Studies. Basically, GWAS is a large-scale analysis that aims to associate genetic mutations with common diseases. The way that a GWAS study is set up is like this. A large cohort is established, many samples are taken from people, and matched for confounding variables like race, sex, and location. The samples undergo SNP genotyping. These results are then analyzed to determine association signals. This step identifies every mutation that infected individuals from the cohort share. These association signals are biologically validated, and the mechanism that determines susceptibility or immunity to infection is identified.
Several factors influence the dynamics of host-pathogen interactions, including length of exposure, morbidity, and geographical spread of the disease, and all of these play a role in determining the accumulation of variants that confer genetic resistance within modern-day populations. If you look at this figure, you can see the timeline of pathogen emergence in human history. When we talk about an old disease, we mean really old, 100,000 years old to be approximate. Now let's take these diseases, from oldest to newest, and do a GWAS to find out what variants play a role in disease resistance. This figure depicts 5 plausible scenarios and what kind of power a GWAS has in each. Several factors either strengthen or decrease the power of GWAS, including time frame, how old the disease is, geographic spread, is it found on every corner of the earth or in a specific location, and how specific it is to a population, whether disease is found in a wide variety of people or in one population. According to this figure, a GWAS of a relatively recent disease that is taken from a specific selected population of infected individuals, like cholera, for instance, has the most power in determining the genetic mutations associated with the disease. In a GWAS scenario on an ancient widespread disease, such as malaria, where the populations are unselected, the results tend to be highly variable as selection is harder to detect when it is shared between populations. Even in a scenario such as leprosy, where the population is specific but unselected, the power of GWAS is variable at best. Lastly, it's important to note that in a case where the disease is very new, not limited to a single population or geographic location, and in the case of HIV, highly mutable, there is no GWAS power because the disease hasn't had enough time to select on a human population. Identifying resistant variants through GWAS is important because if the resistant variants are on the same loci in both an infected and uninfected population, they have the potential to help scientists better understand the disease and possible treatment.
Table 1 shows some diseases on which GWAS has been performed. Malaria is a blood disease that originated in human populations over 100,000 years ago and predates human dispersal from Africa. Theoretically, natural selection from the disease will be reflected in all populations of human beings living today and is a viable target for GWAS. The GWAS of malaria identified SNPs related to blood type and red blood cells. The SNP's relation to blood cells, malaria's target, gives further support to the GWAS results. HIV is a modern disease, but scientists believe SIV, a chimpanzee infecting virus related to HIV, has been passed to humans from chimps at least 10 separate times in the last 100 years. This suggests ancient viruses related to HIV could have selected upon ancient human populations, especially those in close contact with chimps.
What does this have to do with us? Just about everything. These diseases are super common, and the pathogens we are fighting against are in an arms race against us. As we evolve, so do they. The pathogens present in cholera have selected against blood types that contain antigens, inadvertently selecting for blood type O. For everything that a pathogen is doing to us, we are evolving to fight it. This vicious cycle offers unique opportunities to present biological responses to our surroundings.